Good afternoon to all the students of 11th and 12th standard BPC of AP and Telangana. So yes, here from NEET World we are already started with the IPE classes beta, wherein complete detailed sessions of 60 days IPE classes are being taken, where each question is solved in detail. Okay, how you have to attempt each question for more marks, what chapters you have to concentrate, what are the very important questions that are repeatedly appeared in your examination, and most importantly how to remember each answer and in what way if you present you will get more marks. Okay, targeting these things, highlighting these things, 60 sessions of IPE classes are being taken, and a book is launched which is completely with free of cost. Okay, completely the book, IPE book, which is released from the NEET World for each subject is free of cost, wherein in this book very important questions will be covered, complete blueprint with weightage chart is given, and easy simple way of understanding and solving the questions. Previous year exam papers are given and guest model papers are also given in this book. Okay, so look into it more detailedly, and this book is completely for free of cost beta, available to all the students. How you can download this book and how you can get a soft copy of this book, I will tell you on Monday. Okay, so now today we are going to start the second year IPE session, wherein first we are going to target the long answer type of questions. Okay, first long answer type of questions where we can accumulate so many marks from long answer type of question. This is a major domain where you can score high marks, right? So the long answer type of questions from plant physiology, the major chapters to be focused are what? Photosynthesis in higher plants and respiration in plants. Frequently asked questions from photosynthesis in higher plants, if you see, first question, that is explain Kelvin cycle. Kelvin cycle, which is also known as what beta? C3 cycle. So Kelvin cycle in detail, it is asked in many years. If you see, Kelvin cycle is asked in many years, like in March 23, the question number is 19, August 22, May 22, March 18 and March 2016. These many years the question is repeated. Okay, the question is what? About Kelvin cycle. And from Kelvin cycle, if you see Kelvin cycle or C3 cycle, be it from bioenergetics or the enzymatic reactions, many a times we got questions both in NEET and EAP set. So the concept that is C3 cycle or Kelvin cycle from this chapter is important, not just for IPE but also for NEET and for EAP set. Look into the way of attempting the question first. Who discovered the Kelvin cycle? That you have to write it first beta. It is Kelvin and his co-workers discovered a cyclic pathway of carbon dioxide fixation in C3 plants. Okay, in C3 plants it is known as what? Kelvin cycle or C3 cycle. Okay, wherein the first stable product that is formed is 3-carbon compound, that is phosphoglyceric acid. Okay, that is phosphoglyceric acid. Fine, right ma. So here you see Kelvin and co-workers, they have discovered a cyclic pathway of carbon dioxide fixation, that is in C3 plants. This is also known as Kelvin cycle or C3 cycle. Why is it also known as C3 cycle? Because the first stable product that is formed is 3-carbon compound, that is phosphoglyceric acid. This is the introductory, these are the introductory points where the examiner will be looking for mostly. Okay, these are the introductory points to be covered. Now the next step what you have to explain in your answer is the cycle takes place in 3 phases. Okay, 3 phases. First is what? Carboxylation, second is reduction, third is regeneration. Okay, carboxylation, reduction and regeneration. First you see carboxylation. Okay, carboxylation is also known as carbon dioxide fixation. Okay, carboxylation or carbon dioxide fixation, this is the most important and most crucial step of Kelvin cycle. Okay, most crucial step of Kelvin cycle is carboxylation or carbon dioxide fixation, where the first stable carbon dioxide is fixed into a stable compound, where the acceptor is RUBP, ribulose bisphosphate carboxylase oxygenase. That is the enzyme. Okay, Rubisco is the enzyme, whereas the acceptor molecule for carbon dioxide here, carbon dioxide is accepted by a 5 carbon compound, and that 5 carbon compound is ribulose bisphosphate, and the enzyme is ribulose bisphosphate carboxylase oxygenase. That is, Rubisco enzyme is the enzyme that is catalyzing the carboxylation in C3 plants, and the product formed is phosphoglyceric acid. This is the product that is formed. So here you see, all the time you remember this keenly, that the reactions should be written, enzymes should be mentioned properly. Okay. So in C3 cycle, the enzyme which is mediating this reaction is Rubisco, ribulose bisphosphate carboxylase oxygenase. And here if you see the acceptor is 6 RUBP, 6 molecules of ribulose bisphosphate will be accepting 6 molecules of carbon dioxide, and in the presence of the enzyme, that is Rubisco, they will be forming what? 12 phosphoglyceric acid molecules. Okay, 12 phosphoglyceric acid molecules. This is the step. If you are mentioning only for single carbon dioxide, then you have to write as 1 RUBP. Like if you are mentioning only for single carbon dioxide also is fine, where RUBP plus carbon dioxide in the presence of the enzyme Rubisco, fine, you have in the presence of the enzyme Rubisco, 2 phosphoglyceric acid is formed, as this is 5 carbon and this is 1 carbon. Okay. After carbon dioxide fixation, 2 molecules of 3 carbon compound are formed. Okay. 2 molecules of 3 carbon compound are formed, that is 2 phosphoglyceric acid. So here if you see in carboxylation of Kelvin cycle, that is carbon dioxide fixation, which is the most crucial step of Kelvin cycle. This is the most important step where carbon dioxide is fixed into the stable compound, and the acceptor here is ribulose bisphosphate, that is 5 carbon compound, which will accept the carbon dioxide, and the catalyzation will happen in the presence of the enzyme Rubisco, which is the world's abundant enzyme. This point if you mention also it is fine, that is world abundant enzyme, and the first stable product formed is 3 carbon compound, that is phosphoglyceric acid. Any of the reactions you can write beta. If you are taking 6 carbon dioxides and 6 RUBP, then the product is 12 phosphoglyceric acid. If you are considering only one single carbon dioxide fixation, 1 RUBP, 1 carbon dioxide, in the presence of Rubisco it will form like 2 molecules of 3 phosphoglyceric acid. This is the thing that you have to fix out. Next is reduction. Reduction or formation of sugars. Phosphoglyceric acid is reduced to what? Glyceraldehyde 3-phosphate in the reduction step. Remember that. Okay, here phosphoglyceric acid is reduced to glyceraldehyde 3-phosphate. Okay, and here this requires both ATP and NADPH. For single carbon dioxide fixation, in this step 2 molecules of ATP, 2 molecules of NADPH are required. But for 6 carbon dioxide fixations or 6 Kelvin cycles, it requires how many? Total 3 ATP, 6 ATP and 6 NADPH. For 6 carbon dioxide fixation or 6 cycles in the reduction step, total how many are required? 6 ATP and 6 NADPH. So 2-step enzyme action here, one is phosphoglycerokinase and second is G3P dehydrogenase. These are the 2 enzymes that are involved in the reduction step. In each step of Kelvin cycle, enzyme is very important, mentioning of crystal clearly the enzymes. Second is what is bioenergetics, like how much ATP and NADPH are used. And third most important segment is what are the products that are formed. After carboxylation it is 12 phosphoglyceric acid, after reduction it is 12 glyceraldehyde 3-phosphate, where phosphoglyceric acid is reduced to glyceraldehyde 3-phosphate. And here you see in reactions of reduction step if you see, so here 12 molecules of PGA, phosphoglyceric acid, with 12 ATP in the presence of the enzyme phosphoglycerokinase, this forms 12 bisphosphoglyceric acid plus 12 ADP and 12 inorganic phosphate. Okay, 12 bisphosphoglyceric acid along with 12 NADPH in the presence of the enzyme glyceraldehyde 3-phosphate dehydrogenase. This forms 12 glyceraldehyde 3-phosphate, 12 NADP and 12 inorganic phosphate. This will be formed. Clear, right? So, these are the two steps that you have to remember for sure. And in the reduction step, what are the two enzymes that are there beta? Phosphoglycerokinase and glyceraldehyde 3-phosphate dehydrogenase. If you are considering for one carbon dioxide fixation, as I told you for in the carboxylation reaction, at the end of carboxylation, when you are taking one carbon dioxide, you will get what beta? 2-phosphoglyceric acid, right ma? 2-phosphoglyceric acid is formed. This 2-phosphoglyceric acid will require 2 ATP, and in the presence of the enzyme phosphoglycerokinase, it forms 2-bisphosphoglyceric acid and 2 ADP and inorganic phosphate. Okay. And second is 2-bisphosphoglyceric acid, 2-bisphosphoglyceric acid plus 2 NADPH, 2 NADPH in the presence of the enzyme glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase, it forms 2 NADP and 2 inorganic phosphate. This will be formed at the end. Okay. So, any reaction is accepted beta. You can also take one carbon dioxide fixation, or you can take 6 carbon dioxides or 6 cycles of Kelvin, but mention clearly the energetics. Okay. If it is 1 carbon dioxide, it has 2 ATP, 2 NADPH, and products also should be clear. If it is 6 carbon dioxide, it is 12 ATP, 12 NADPH, and products also should be clear. So, at the end of reduction, what you are getting ma? 2 glyceraldehyde 3-phosphate. So, here you mention this point keenly. Out of 12 phosphoglyceric acid, 2 will form what? Glucose, and 10 molecules will go to regeneration. This point is very important ma. Out of 12 glyceraldehyde 3-phosphate, how many will go to formation of glucose? 2 molecules of G3P will go for the formation of glucose, and 10 molecules will go for the formation of regeneration. Okay, regeneration of RUBP. And I have given you a mnemonic to remember the two steps here carefully, that is ATP and NADPH reduce PGA to sugar. Okay, this is the mnemonic. You need not to mention mnemonic in your answer again. See, this is mentioned, mnemonics everywhere in the book are mentioned for your easy remembrance of the answer. Okay, not to present in the exam. Okay. Next, if you see ma, regeneration. The key role regeneration is 6 RUBP will form from 10 glyceraldehyde 3-phosphate. This involves a complex sequence of sugar interconversions. Okay. So, it involves 7 steps of reactions in the regeneration. Okay, where 10 molecules of glyceraldehyde 3-phosphate can give what? 6 phosphoglyceric acid, wherein one molecule of G3P has how many carbons? 3 carbons. Okay. Remember that carefully. Now, in regeneration, if you see, the sequence of regeneration will have these many reactions. First is isomerization, second is aldol condensation, third is dephosphorylation, fourth is transketolation, fifth aldol decondensation, sixth dephosphorylation, seventh is transketolation, 8 epimerization, and 9 is isomerization, 10 phosphorylation. So, there are 10 steps in regeneration. How you can remember these 10 steps? For this, mnemonic is very essential. Okay. The 10 steps you can remember with the mnemonic. See, all the enzymes. I am friendly teacher, always sharing smart tricks everywhere in return. Okay. This is the mnemonic which I gave for remembering the 10 reactions in the regeneration step. First, I, if you see, first you note down the mnemonic and then we will get into the details of each reaction. I, you see here, I is what? Isomerase, fine. A is aldolase, F friendly, fructose 1,6-phosphatase, T is transketolase, A is aldolase, S pseudohepulose 1,7-bisphosphatase, next S, you see, that is transketolase, T, sorry, not S, this is T transketolase, E epimerase, and I here isomerase, lastly R RUBP kinase. Okay. These are the steps that you can remember just with one mnemonic. I am friendly teacher, always sharing smart tricks everywhere in written. Isomerase, aldolase, fructose 1,6 phosphatase, transketolase, aldolase, ceroheptulose 1,7 bisphosphatase, transketolase, epimerase, isomerase and RUBP kinase. These are the steps. Okay, in this way you can remember the sequence easily. Now we will go with the details of each reaction. Each reaction details you see. Yeah, isomerization, here you see 4-glyceraldehyde 3-phosphate after isomerization. See, 4-glyceraldehyde 3-phosphate after isomerization they will turn to 4-dihydroxyacetone phosphate. Okay, 4-dihydroxyacetone phosphate. 4-dihydroxyacetone phosphate. Next is dephosphorylation, that is 2-fructose 1,6 bisphosphate in the presence of the enzyme fructose 1,6 phosphatase. This forms 2-fructose 6-phosphate and 2-inorganic phosphate. And then you see transketolation. Transketolation 2-fructose 6-phosphate and 2-glyceraldehyde 3-phosphate, which is over here. Yeah, 2-glyceraldehyde 3-phosphate. This in the presence of the enzyme transketolase. Transketolase, this forms 2-xylulose 5-phosphate and 2-erythrose 4-phosphate. Okay, 2-xylulose 5-phosphate and 2-erythrose 4-phosphate. Next is aldol condensation. Aldol condensation, here you see 2-dihydroxyacetone phosphate and 2-erythrose 4-phosphate in the presence of the enzyme aldolase. This forms 2-ceroheptulose 1,7 bisphosphate. This is 7-carbon compound, okay, which is the largest compound that you can see in the regeneration. So it is ceroheptulose 1,7 bisphosphate. Next, after aldol condensation from ceroheptulose 1,7 bisphosphate you see. Next step is what? Dephosphorylation. In dephosphorylation what happens ma? 2-ceroheptulose 1,7 bisphosphate in the presence of the enzyme ceroheptulose 1,7 bisphosphatase. This forms 2-ceroheptulose 7-phosphate and 2-inorganic phosphate. And next step is what? Transketolation. Transketolation where ceroheptulose 7-phosphate and 2-glyceraldehyde 3-phosphate in the presence of transketolase. This forms 2-xylulose 5-phosphate and 2-ribose 5-phosphate. Okay, both are 4-carbon only. Ribose is 5-carbon and cellulose is 4-carbon. Next reaction is epimerization, where cellulose 5-phosphate in the presence of epimerase. This forms 4-ribulose 5-phosphate. Okay, and then isomerization, 2-ribose 5-phosphate in the presence of isomerase. This forms 2-ribulose 5-phosphate, after which phosphorylation, where 6-ribulose 5-phosphate in the presence of 6-ATP and the enzyme RUBP kinase. This forms 6-ribulose 1,5 bisphosphate and 6-ADP. 6-ADP, whenever ADP is there you can also include 6-inorganic phosphate. Okay, these are 10 sequential steps that you can go through in the regeneration step. And to remember the enzymes of these 10 steps, I have already given you the mnemonic. Okay, already the mnemonic is given with which you can remember the regeneration steps. So with this the complete details are clear, Noma? Carboxylation, reduction and regenerations. Whatever we have taken down here for the reactions, it is for 6-carbon dioxide fixation. As such, the energetics and the products are given. Here you see bioenergetics of Kelvin cycle. Fixation of 6-carbon dioxide with 6 RUBP via Rubisco. Okay, 6 carbon dioxides with 6 RUBP via Rubisco, and there is no direct ATP or NADPH that is used in this step. The products that are formed are 12 molecules of 3-phosphoglyceric acid. Next reduction you see 12 phosphoglyceric acid turns to 12 bisphosphoglyceric acid and then 12 glyceraldehyde 3-phosphate. After which you see this consumes how much? 12 ATP and 12 NADPH. Out of 12 glyceraldehyde 3-phosphate, 2 exit to form what? 1 glucose and 10 are used in regeneration. In regeneration finally you see 10 glyceraldehyde 3-phosphate molecules are regenerated to 6 RUBP by consuming how much? 6 ATP. This ensures continuity of the cycle. Again this is the major point. Which step out of 3 steps in the Kelvin cycle will help the cycle to continue? That is regeneration. Okay, and this is the mnemonic that is used for the enzyme sequence. Fine, right? So complete answer about Kelvin cycle is clear, Nama? Any doubt you are having, so you can drop a message in the comment box. I will be answering your doubts. Any doubt you can answer. Continuation lo undi. Here, second question you see, this is the most important question, n number of times asked. Okay, this is on cyclic and non-cyclic photophosphorylation. You see, ma, in the light of modern researchers, describe the process of electron transport, both cyclic and non-cyclic photophosphorylation. First, electron transport system in photosynthesis. You have to explain what is electron transport system. In photosynthesis, light excites the electrons in chlorophyll molecules which are transported through electron transport chain to regenerate both ATP and NADPH. Two photosystems are involved over here, that is photosystem 2, which has highest excitation is P680, photosystem 1 that is P700, highest wavelength of excitation is P700. So first you have to mention what, ma? Light excites the electrons in chlorophyll molecules which are transported through electron transport chain. This will regenerate both ATP and NADPH. So here you see first electron photosystem 2. Photosystem 2, this is discovered by whom? Robert Hill. This is discovered by Robert Hill in the year 1939, where electron transport chain is explained in detail. Here light excites P680 and electrons are transferred to the primary acceptor. Primary acceptor is pheophytin. Examiner will be looking for the wavelength of each photosystem and what is the primary acceptor, what is the last acceptor and what is the sequence of the protein molecules. So here you see water molecules will split where photolysis will occur from photosystem 2 only, and here you also mention the location. It is not mentioned over the PDF here, but then please mention the location in your exam as well. Water molecule split, that is photolysis, happens in the inner membrane of the thylakoid, inner thylakoid membrane towards thylakoid lumen, towards thylakoid lumen. Fine. This occurs in the inner thylakoid membrane towards thylakoid lumen, where photolysis of water takes place. 2 water molecules are photolysed to 4 protons, oxygen and 4 electrons. This is how they are split into inside the thylakoid lumen. So in the inner thylakoid membrane there is a complex called as oxygen evolving complex, OEC. That is what, ma? Oxygen evolving complex. Oxygen evolving complex, that is situated attaching to the inner thylakoid membrane towards the thylakoid lumen, where photolysis of water occurs. Here electrons will replace the lost ones in P680. In photosystem 2, the complete sequence of the electron is mentioned. First is pheophytin and then plastoquinone, pheophytin, plastoquinone, followed by cytochrome complex, that is cytochrome B6 complex, B6F complex, and then you can write cytochrome B6F complex followed by plastocyanin. What is the last electron acceptor of photosystem 2? That is plastocyanin. Then it is sent to photosystem 1. Here photosystem 2 P680, which is discovered by whom beta? Robert Hill 2. And here this is electron transport chain is explained, where the light excites up to what maximum wavelength here in photosystem 2? P680, and the electrons are transferred to primary acceptor, that is pheophytin. Here water molecules will split where photolysis happens in the inner thylakoid membrane. Attach it to thylakoid lumen. What complex is present? Oxygen evolving complex. Fine, right? And this is the flow of the electrons. Electrons replace the last ones in P680 like pheophytin, then plastoquinone, cytochrome B6F complex, and the last electron acceptor is plastocyanin, which thereby sends the electrons to photosystem 1. Now you see, here you can remember this electron flow with a mnemonic. I am again saying mnemonics are just to remember the answer, not to be presented in the exam. The mnemonic you see here, water pushes P680, pheo quickly cycles to plastocyanin, okay, pheophytin, cytochrome B6 complex, and then plastocyanin, okay. Water pushes, that is photolysis of water, pheophytin and cytochrome B6F complex, plastocyanin, all are mentioned in this, okay. Mostly mnemonic is not necessary here, but to the students who can remember it easily, for them I have mentioned, okay. It is not required for all the students, mnemonics is not required for all the students, they can directly remember the answer, but to the students who has little, like they need some mnemonic or some helper to remember the cycle, for them the mnemonics are given, okay. And mark my words, again and again I am stressing out, mnemonics need not to be presented in the exam, that is only for your memory purpose. Next is photosystem 1. Photosystem 1 maximum velocity is P700, which absorb light electrons and then it passes to ferredoxin complex. Here electrons from photosystem 2 will replace P700, okay, replace P700 losses, and here ferredoxin component which is in photosystem 1, this gives NADP reductase and this forms NADPH, okay. And here NADPH is used in Kelvin cycle in the reduction phase. In the reduction phase, NADPH is used in the Kelvin cycle. Fine, right? The NADPH which is released out in the P700 is used in the Kelvin cycle, and the mnemonic you can remember over here is P700 fires. F is what? Ferredoxin, fires electrons fast to NADPH, okay. And here by the enzyme NADP reductase, NADPH is formed. The final electron acceptor is NADP, okay. Here you can see ferredoxin to NADP, and here NADP is the final electron acceptor in the non-cyclic photophosphorylation. Remember that. If you mention in your answer when cyclic and non-cyclic photophosphorylation is asked, if you are mentioning the primary acceptor and the last acceptor of each photosystem, then you will get an additional points, okay. So in photosystem 2 the primary electron acceptor is pheophytin, last is plastocyanin, and in photosystem 1 if you see, the electrons will be accepted by ferredoxin, and the last electron acceptor of non-cyclic photophosphorylation is NADP. NADP plus is the last electron acceptor. By the enzyme NADP reductase, NADPH is formed. NADPH is formed towards the stroma of the chloroplast, not in the thylakoid membrane. NADPH is formed where ma? In the stroma of the chloroplast. Here you see the detailed diagram, as it is you have to remember ma. This diagram as it is you have to remember, I am telling you. Once again clearly I will explain you, listen, okay. Non-cyclic photophosphorylation as it is, this diagram you have to remember and you have to present, okay. First light reaction which happens in 3 steps. First is what? Absorption of light in the form of photons. Second is photolysis of water. Here you can see associated with photosystem 2 there is a complex called as oxygen evolving complex towards the thylakoid lumen, where the water is photolysed, H2O is photolysed to H plus, single water molecule if you take 2 H plus oxygen atom and then 2 electrons. Okay, if you take 4 water molecules, then complete oxygen molecule will be formed. So here after absorption of light in the form of photons, it gets excited. Maximum wavelength of photosystem 2 is what? P680, and the primary electron acceptor is primary electron acceptor. Primary electron acceptor is your pheophytin. The next it is plastoquinone. Plastoquinone has special quinones, that is QA and QB. This is called as quinone pool. Then cytochrome B6F complex, cytochrome iron sulfur protein complex, this is called as cytochrome B6F complex, after which the electrons are pumped to plastocyanine. Plastocyanine is the last electron acceptor of photosystem 2. Now from here the electrons are submitted to photosystem 1, where from photosystem 1 maximum excitation is P700. Okay. From here the electrons are again passed on to ferrous sulfur complex. Electrons are passed on to ferrous sulfur complex, after which electrons are submitted to ferredoxin, and the ultimate or last electron acceptor over here is NADP+. NADP+ is the ultimate or last electron acceptor, and then it is converted to NADPH by the enzyme NADP reductase. By the enzyme NADP reductase. This is the non-cyclic photophosphorylation complete sequence. Clear, right? Easy? Now, photophosphorylation, that is cyclic photophosphorylation. Here cyclic photophosphorylation, this is the process of synthesizing ATP from ADP and inorganic phosphate in the presence of light. Okay. This is using energy which is derived from the movement of electrons through photosynthetic electron transport chain in the chloroplast. This occurs light dependent reactions of photosynthesis. Okay. This takes place only in the light dependent reactions, and the introductory paragraph is very important even for cyclic photophosphorylation. So here if you see for cyclic photophosphorylation, only one photosystem is involved, and for this photosystem maximum wavelength of light is what? P700. One point to be highlighted in the cyclic photophosphorylation is there is no photolysis of water. No photolysis of water, and the photosystem 1 is associated in the stroma lamellae. Stroma lamellae, this is the major point to be dealt. They are present in the stroma lamellae. Photosystem 1, ferrous sulfur complex, ferredoxin. Here ferredoxin, when it is transmitting the electrons to cytochrome B6 complex, ATP is pumped out, and from cytochrome BC to cytochrome F, one more ATP is pumped out. So the ultimate ATP release from photosystem 1 or cyclic photophosphorylation is just 2 ATP. Fine, right ma? And then the ultimate electron acceptor over here is plastocyanine. The first electron acceptor of cyclic photophosphorylation is ferredoxin, and the ultimate electron acceptor is plastocyanine, where the electrons are again submitted to photosystem 1. This is the cyclic chain. Here what are the highlights that you have to mention? That no photolysis of water. Photolysis of water is not associated with cyclic photophosphorylation because only PS1 is involved. And where is PS1 situated? In the stroma lamellae. Photosystem 1 is situated in the stroma lamellae, and this is cyclic photophosphorylation. Clear, right ma? Clear with this? Lastly, the tabulated form of cyclic and non-cyclic photophosphorylation. Cyclic photophosphorylation, firstly you see only photosystem 1 is involved in the cyclic photophosphorylation. In non-cyclic though 2 are involved, both photosystem 1 and photosystem 2, both are involved. Both photosystem 1 and photosystem 2 are involved. And in the cyclic photophosphorylation, which is cyclic Electrons are released by PS1 and they will return back to the PS1. In non-cyclic photophosphorylation, electrons move from water to PS2 and then to PS1 and then to NADP plus, where NADP plus is the ultimate electron acceptor. Lastly, only ATP is produced here. In non-cyclic photophosphorylation, if you see ATP, NADPH and oxygen will be evolved. In cyclic photophosphorylation, there is no photolysis of water. In non-cyclic, photolysis of water will occur at PS2 releasing out oxygen. In cyclic, which is not formed, and here water molecule is formed, fine, and this provides additional ATP that is needed for Kelvin cycle. Here this provides both, in non-cyclic both ATP and NADPH for carbon fixation, and oxygen is also released as byproduct. One more point you can highlight over here, here this happens mostly in the anoxygenic photosynthesis, this happens in the cyclic photosynthesis, fine, right. This happens in the cyclic photosynthesis, this happens in non-cyclic photosynthesis, where cyclic photophosphorylation, only cyclic photophosphorylation occurs in oxygenic photosynthesis, oxygenic photosynthesis. Non-cyclic, cyclic occurs in anoxygenic photosynthesis, non-cyclic in oxygenic photosynthesis, as oxygen is evolved as byproduct, oxygenic photosynthesis. And lastly, what you have to mention is this occurs in stroma lamellae, as PS1 is situated in the stroma lamellae, and here non-cyclic, this occurs in thylakoid lamellae, okay, thylakoid lamellae, which occurs in thylakoid lamellae where non-cyclic photophosphorylation occurs, where both PS1 and PS2 are present. With this, the complete description of two answers majorly n number of times asked from photosynthesis. One is your Kelvin cycle and second is cyclic and non-cyclic photophosphorylation, which is clearly dealt, what are the points to be highlighted, how you have to frame out the question, and the mnemonics are given to remember all the enzymatic reactions and the electron sequences in a sequential manner. With the help of mnemonics you can remember them, but again and again I am telling you, do not mention the mnemonics in your exam paper, okay. Just go through this and then we will start with respiration in plants. Next, respiration in plants. Here, this is the major important question. Give an account of glycolysis. Glycolysis n number of times asked in your previous IPE papers, and glycolysis is the major hotspot where the bioenergetics are targeted, where the electron transport, like the sequence of the enzymes is targeted in all the 10 steps, be it in payoff phase or in the preparatory phase. All the steps are highlighted, targeted with enzymes and bioenergetics, not just for IPE but also for your NEET and MCET. We will exercise the concept all together, okay. So, give an account on glycolysis, where does it occur, what are the end products, trace the fate of these products in both aerobic and anaerobic respiration. This question has appeared in your previous papers like in June 2023, August 2022, March 2020, May 2018, May 2017, May 2015 and March 2015. These many years in your previous papers of IPE, this question has appeared. So, here if you see in the answer, what is that you have to mention in the beginning. Glycolysis is the process of partial oxidation of glucose or other hexoses, where partial oxidation of glucose 6 molecules of glucose will be converted to 2 molecules of pyruvic acid. You can also mention it as pyruvate with the production of what ma? ATP and NADH. There in the cyclic and non-cyclic photophosphorylation, you will be using the terms like NADPH. But here in the glycolysis, you have to mention out that it is production of both ATP and NADH, not NADPH. It occurs in the cytoplasm of living cells. This occurs in the cytoplasm of living cells, and this functions as first step of both aerobic and anaerobic respiration. Okay, this functions as the first step of both aerobic and anaerobic respiration. You can also write in this way. This is the common pathway for both aerobic respiration and anaerobic respiration. In this way also you can present it. Okay, and then this pathway was first described by whom? Emden, Meyerhoff and Parnas. It is Emden, Meyerhoff and Parnas. Hence it is also known as EMP pathway. Okay, this is also known as EMP pathway, and this oxidation is also termed as partial oxidation of glucose or incomplete oxidation of glucose. Incomplete oxidation of glucose. Incomplete oxidation of glucose. In this way also you can present it. So, in the beginning of this question you have to write the major points of the introductory points, and then you have to mention like what pathway it is, that is the EMP pathway. Next you see pathway of glycolysis. Pathway of glycolysis. In plants, sucrose is first hydrolyzed into glucose and fructose. Here sucrose is first hydrolyzed to what? Glucose and fructose by the enzyme invertase. Okay, this is by the enzyme invertase. Both monosaccharides, then they will enter into what? Glycolysis. Like how glucose molecule, we know that food is stored in the form of glycogen in the animal cells, starch in the plant cells. But for the transport of the sugar, it will be transported in the form of sucrose. Sucrose, once it enters the cell, sucrose once it enters the cell, this sucrose is cleaved to glucose and fructose. Glucose and fructose by the enzyme invertase. Fine, this is by the enzyme invertase. Both monosaccharides, then they will enter into glycolysis. The pathway consists of what? 10 enzymatic steps, which can be divided into 2 phases. First phase is preparatory phase, that is 1 to 5 steps. This is energy investment, where the energy investment occurs. Here in the preparatory phase, ATP will be consumed. ATP is consumed in the preparatory phase. Next is what is payoff phase, from 6 to 10 steps. Here energy is generated. This is energy generation phase, where both ATP and NADH are produced, not NADPH over here. Here ATP and NADH are produced. So, this is what you have to mention now. Introductory phase and then pathway of glycolysis, and then you have to mention about both the phases, that is preparatory phase and payoff phase, energy investment phase and energy generation phase. First you see the stepwise reactions of glycolysis. First is what ma? Phosphorylation. Phosphorylation, where glucose is first phosphorylated to glucose 6-phosphate by the enzyme hexokinase. Remember that this is by the enzyme hexokinase. This utilizes single ATP. This utilizes how much ATP? 1 ATP. Second step is isomerization. Okay, second step is what beta? Isomerization, where glucose 6-phosphate is isomerized to fructose 6-phosphate, and the enzyme here used is phosphoglucoisomerase enzyme. Third step, which is the rate limiting step, most important step of glycolysis, that is fructose 6-phosphate is converted to fructose 1,6-bisphosphate. This is by the enzyme phosphofructokinase, which is the rate limiting enzyme, which is the major enzyme in the glycolysis, that is phosphofructokinase. So, the first 3 steps are clear beta. First is phosphorylation, second is isomerization, and third is second phosphorylation. and the enzymes of each step must and should be noted, and the products should be highlighted. This enzymatic machinery is not just important for IPE, again and I am telling you, this is a major concept, hotspot concept for both NEET and MCET. So, look into it with major interest and go through it. Okay. Next, first 3 steps are clear. Fourth step is what? Cleavage. Okay, wherein 6 carbon fructose 1,6 bisphosphate will be cleaved to glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. Okay, it will be cleaved to 6 carbon fructose 1,6 bisphosphate is cleaved to 3 carbon glyceraldehyde 3-phosphate and 3 carbon dihydroxy acetone phosphate by the enzyme aldolase. Next is what? Isomerization, where dihydroxy acetone phosphate is isomerized to glyceraldehyde 3-phosphate. This is by the enzyme triose phosphate isomerase. DHAP is also 3 carbon, glyceraldehyde 3-phosphate is also 3 carbon, and this is isomerized by the enzyme triose phosphate isomerase. Thus, 2 molecules of G3P are obtained from 1 molecule of glucose. From here, from 6th step onwards, you can calculate the energetics for 2 molecules of G3P. You can calculate the energetics for one total glucose molecule, as DHAP is again getting isomerized to G3P. Okay. Now, here you see 6th step, that is oxidation and phosphorylation. Where G3P is first oxidized to 1,3-bisphosphoglyceric acid, and here phosphorylation is also done. This is by the enzyme glyceraldehyde 3-phosphate dehydrogenase. The enzyme used over here is G3P dehydrogenase, and this will produce out what? NADH. Next, ATP formation. If you observe ATP formation over here, this is known as substrate level phosphorylation, where phosphate group is added to ADP directly from the substrate. Okay. Phosphate group is added for ATP formation. Here ATP is produced and phosphate group is added here directly from the substrate. That is why this is called as what? Substrate level phosphorylation. What is substrate level phosphorylation? Which is step beta? 7th step is for substrate level phosphorylation, where 1,3-bisphosphoglycerate is phosphorylated to is turned to 3-phosphoglycerate and ATP is produced here. The enzyme is phosphoglycerate kinase. This is the enzyme. Okay, phosphoglycerate kinase. And next is mutation reaction. Here 3-phosphoglycerate is produced to what? 2-phosphoglycerate. This is by the enzyme phosphoglyceromutase. 9th step, which is dehydration step, where 2-phosphoglycerate is dehydrated to phosphoenol pyruvate. Okay, 2-phosphoglycerate is turned to what? Phosphoenol pyruvate, and the enzyme is what? Enolase enzyme over here. Next is final ATP formation, that is phosphoenol pyruvate is done to pyruvic acid. The end product formed is pyruvic acid. This is by the enzyme pyruvate kinase. This produces one ATP molecule. Net energy yield of glycolysis per 1 glucose. Per 1 glucose, what is the net energy release? Net gain of ATP used is what? 2 ATP. Gross ATP formed is what? 4 ATP. NADH formed at the 6th step. 2 NADH are formed, and here total ATP formed is 8 ATP. How you are calculating this? First you see 2 ATP. Net gain of ATP is 2 ATP. Gross ATP formed is 4. That is at 7th step and 10th step of glycolysis, 2 ATP are produced at each step, right? 2 ATP are produced, one at the 7th step and one at the 10th step. So, gross ATP released is 4 ATP. Of this 4 ATP released into the cytoplasm, 2 ATP will be utilized at the preparatory phase in first and third step. Excuse, 2 ATP are utilized. Fine, right? In the first and third step, 2 ATP are utilized. So, the net gain of ATP is how much? 2. Fine, right? Next, NADH formed. NADH is formed at 6th step, that is 2 NADH. Now we need to calculate the total ATP that are formed. Net gain is 2 plus 2 NADH. 1 NADH is equal to 3 ATP. Fine, 1 NADH is equal to 3 ATP. 2 NADH is equal to 3 plus 3, that is 6 ATP. 6 ATP plus net gain of ATP is 2, right? 6 ATP plus 2 ATP, on total it is 8 ATP. Fine, right ma? On total it is 8 ATP. This is the bioenergetics, very important, you have to highlight this in the exam. Clear, right? Next, fate of pyruvic acid. What is the fate of pyruvic acid? This also you mention. Okay, pyruvic acid enters mitochondria if it is aerobic condition. So here glycolysis, what is the site of glycolysis ma? Okay, in cytoplasm glycolysis occurs, wherein glucose is turned to 2 molecules of pyruvic acid. Okay, 2 molecules of pyruvic acid. This pyruvic acid will directly enter into the mitochondria if it is aerobic respiration. Where it will enter? Directly into the mitochondrial matrix of mitochondria. Okay, this will directly enter into the mitochondrial matrix of mitochondria, wherein pyruvic acid enters mitochondria and is oxidized through Krebs cycle and electron transport chain. So once after entering into the mitochondrial matrix, it will do what? Krebs cycle and then electron transport chain. ETC, electron transport chain, occurs in the inner mitochondrial membrane. So the end products that are formed after aerobic respiration is carbon dioxide, water, and 36 ATP are formed in a eukaryotic cell. Next, in anaerobic condition, in muscles though, lactic acid fermentation will take place. Okay, in muscles lactic acid fermentation will take place, wherein pyruvic acid is turned to what? Lactic acid by the enzyme lactic acid dehydrogenase. Need not to mention here, but I am telling you, lactic acid dehydrogenase. This causes what? Muscle fatigue. This happens during vigorous exercises. Okay, we will get muscle cramps and all. That is because of the instant ATP release which is done through what? Anaerobic respiration, that is lactic acid fermentation. Next, in yeast cell, alcoholic fermentation is done, wherein pyruvic acid is converted to ethanol and carbon dioxide. Okay, this is converted to ethanol and carbon dioxide by the enzyme pyruvic acid dehydrogenase. This is by the enzyme pyruvic acid dehydrogenase. Basis for what? Brewing and baking. This is the basis for brewing and baking. This is for both aerobic conditions and anaerobic conditions. Clear with this? The fate of pyruvic acid. Next is what ma? Significance of glycolysis. What is the significance of glycolysis here? The significance of glycolysis, you see, this provides quick energy even in the absence of oxygen. Okay, glycolysis will provide quick energy even in the absence of oxygen. This is the universal pathway which is common in both prokaryotes and in eukaryotes. This is common for both prokaryotes and eukaryotes. The intermediates of this pathway are precursors. These intermediates are precursors for amino acids, nucleotides and lipids. Okay, intermediates are precursors for amino acids, nucleotides and lipids. With this, the complete glycolysis answer is clear, right? And the next we have to look into is Krebs cycle. Krebs cycle and ETS. Just a second. Yeah. So Krebs cycle and ETS we will look into the next class ma. Fine. Till glycolysis though it is clear, right? So here in today's session we have exercised 3 long answers where there is a high expectation that we can get one question this year. So those 3 LAQs include what? About Kelvin cycle, that is C3 pathway, second is cyclic and non-cyclic photophosphorylation, and third is what ma? Glycolysis. These are the 3 major questions that we have exercised today. Please go through them carefully. If you are having any doubts from them, you can ask me. These 3 concepts are important not just for IPE but also for NEET and EAP set. Last and finally, I would like to end this session by telling you about the book that is being going to release very soon, and from Monday the soft copies will be available beta. These books are completely for free of cost, completely for free of cost, wherein these books are specially designed by NEET World's faculty, and this is for completely free of cost. Soft copies you can directly get it. The access for this will be given from Monday, wherein this book, well crafted, longs, shorts and very shorts are given, blueprint with weightage chart is given, simple and easy questions to understand, previous year exam papers are also given, guest papers and model papers are given, and a complete 60 days revision slots where each question is detailedly explained in each class. Okay? So, do not miss these sessions at all. Thank you.